it's a pleasure to be here. And indeed, kind of what I was hoping to do today is a little bit to tell you some of the big data and some of the challenges we have as we start thinking about, how do we apply networks and network thinking to the cell? And I should start by saying that I'm a network scientist. I define myself as a network scientist. I started out my career as a physicist. And eventually, about 20 years ago, I started to focus entirely of how do networks form, how do they evolve, and how do we apply the fundamental knowledge that we have about networks to different problems. And in the last kind of 15 years, that problem that has really focused about half of my, engaged about half of my lab has focused on what we call network medicine. And the question is, why network medicine? And we can actually start with this journey. This is actually a cover of Nature that I helped Nature design about 2 years ago when we celebrated the 10-year anniversary of the Human Genome Project. And the reason why actually the kind of the visual that we kind of came out with it is that we tried to show what did the Genome Project really provided us that we did not know before the human genome was released. And at the end, we kind of came up with this image, and I'll explain you what's on this image, where we kind of used essentially the icebreaker idea, where we mapped out what you see here, every circle corresponds to a human gene within the human genome. And the circles particularly are the chromosomes. The individual icebergs are the individual genes. And we mapped out how many papers cited these genes, or how many papers actually mentioned these genes before the Human Genome Project and after. So, and what you see actually is that each iceberg, the height of the iceberg shows how much knowledge was available before the human genome was released on that particular gene. And what's underneath is kind of the new knowledge that the Human Genome has provided us, that was not available when the Human Genome Project came out. So effectively, how many papers mention this particular gene before and after the human genome? And kind of the whole idea of this cover was to show that indeed there's a huge amount of knowledge that was under the water, that was unveiled once we had the tools of the Human Genome Project and the kind of the resources that are provided by the Human Genome Project. And what I would like to tell you today about it is that, me and many of my colleagues actually at Harvard Medical School are entirely focused on human health from the perspective that if we know all the genes and all the mutations within that, then we will understand and we will be able to cure disease. And what I would like to argue today is that that is not enough. That is not enough to know what are the genes and what are the mutations within them. A key piece of the story that is missing is how do these genes or the product of these genes interact with each other, and how the breakdown of that network that results from these interactions really leads to human disease. And what I would like to argue for today is that having the human genome is like having the proverbial phone book. Some of you may not even know how a phone book looked like back then, when we had a big thick book actually on our desk, which provided names and addresses. But in order to really get to find somebody who was on that phone book, was not enough to have the name and address and the phone number. You also needed a map. That is, how do you get there? And network medicine really kind of is the attempt to provide that map. So what I would like to tell you a little bit about is that what are the networks that really matter within the cell, and how do they matter, and how do we use this knowledge actually to understand human disease? So what you see here is nothing but the interactions between the proteins within the cell. So just a little primer to remind us that the genes themselves are typically inert. What their role is to produce different products, and the most important product of the genes are the proteins. And the proteins are the machineries within the cell that get actually things done, but they can never do things alone. They do so by interacting with other genes. And both at Harvard and other places, there were massive efforts in the last kind of 20, 30 years to map out who is interacting with whom. And the product of the journey are maps like that. And my role, or at least my lab's role, is to make sense of these networks and to use these networks to understand, like where they come from and eventually how they lead to disease. Now, to understand what the concept of disease within that network, I'm going to give you an analogy that is a little far-fetched. So I'm going to ask you to kind of suspend disbelief as we do in the movies and assume that what you see here is a map of the cell, right? And so if Manhattan, and here the intersections would correspond to the proteins, and the road segments in this particular example would correspond to the road segments would correspond to the interactions. So very hypothetical. Now, if this would be a cell, then the question is, how would function be kind of distributed within the network? And you know that if you go to New York and you would like to go to a theater, you would realize that theaters are not all over the city, but there's a particular neighborhood around Times Square where most of the theaters are located. And you say the function of the city that relates to theater is located in that particular neighborhood. And then if you kind of say, hey, I'm interested in very high-end art, you would realize that, again, the high-end art galleries are not all over the city, but tend to be concentrated in one particular neighborhood, the former meatpacking district, and now it's kind of known as the art district. And what I want to kind of emphasize in this particular example is that when we look at cities, and that transfers to cell, functions are not distributed uniformly. Some are, but most are not. But there are well-localized neighborhoods where certain functions emerge within the cell, within the city. And the same is true actually within the city, within the cell, that when we look from a network perspective, I offer you evidence for that. When we look from a network perspective, proteins that relate to a certain function don't seem to be anywhere in the network, but tend to interact with each other. They do so because the function is achieved by the interaction of several components. Now, if this is the cell, then the question is, what is a disease? And typically a disease is a breakdown of a function, right? So therefore, if we say that functional proteins are well localized in the network, so should the disease proteins. And what would mean actually in this case that we say, hey, cancer is located on Wall Street, for example, and you put bipolar disorder probably about Times Square, and maybe you can put asthma next to Washington's bridge, where Manhattan breeds much of its mess into New Jersey, right? So in a way, the concept that is developing through this very simple example that we're going to take it back to the much more complicated case is that if you want to understand what is a disease in the network sense, first you need a map, right? And that's a totally experimental process that people here at Harvard, at Dana-Farber, like Mark Vidar's lab and others are involved in. Then you need to find where in the map are the particular neighborhoods that relate to a function and eventually to a disease, and we'll call this disease module. And then you need to perturb it. And in the biological language, this means you have to drug it. You perturb typically by sending a molecule that binds to those neighborhoods, and that's a drug. Now, this is all very nice and very hypothetical. The question, how does this look within the cell? And here I'm showing you the map that I showed you earlier with one particular caveat. So again, I'm kind of every dot here corresponds to a protein. And here I colored proteins that are involved in one particular disease, and that's asthma in this particular case. The reason is that for many years, we've been focusing a lot on kind of understanding asthma from a network perspective. And what you see is that the asthma genes indeed are not randomly distributed in a network, but tend to be located in one particular neighborhood. And a few proteins that involved in asthma kind of are a little further away, but mostly because the network when we did that was very highly incomplete. Now it's actually much more complete. And sometimes a gene could be labeled asthma gene and really may not have a function related to asthma. So it's kind of a mislabeled gene. But fundamentally, what we see is that there is this particular network neighborhood on the right-hand side that's where the kind of the asthma genes are related, just like I showed you on the map earlier. Now, the question is, how do we know that we are right, that there is indeed such a network neighborhood? And that's where we kind of start developing network-based measures. The first thing you do is that you say, what's the largest connected component? The largest connected component in graph theory is really just the largest number of proteins that have at least one interaction to the other, and they form one cluster, right? Now, the problem with this is that if you just throw a certain number of genes onto the network, you will find some clusters. So you would find some of them to be connected to each other. So you start asking, largest connected component, for example, here is the size 11. Is that size could have emerged by chance, or it is really kind of something out of order? And in this particular case, this is not asthma, but multiple sclerosis, we find that 11 is too large to emerge by chance. So that's kind of an indication that indeed we're pointing to the network neighborhood where that particular disease resides. And there are also other measures like the shortest distance, like all the genes who don't connect to this cluster, but they're involved in asthma or in multiple sclerosis. How far are they from that cluster? And indeed, you find that to be closer than what you would expect by chance. And there are other measures as well right now, but fundamentally what the network science perspective provides us is a set of tools to tell us, yes, indeed, that neighborhood that we saw for asthma is really meaningful because there is an agglomeration of asthma genes that you cannot simply explain by chance. So now once you did that, then you would say, who else could be involved? And that's where kind of the algorithmic different type of tools kind of come into play. When we started working on that 10 years ago, these were network-based tools. And what you see actually here in the video is algorithms that kind of look in the neighborhood of the disease genes and say, who else could be a disease gene that is not being labeled as a disease gene? And the hypothesis is very simple. If you interact with many disease genes, you're very likely involved in the disease. And again, what does it mean to interact with many disease genes? Well, again, you use statistical measures to identify, like whether that's meaningful, that number of connections you have. And at the end, you end up with this object that we call the disease module. And the disease module is this kind of nice pink looking thing, which is like a compact module within the network. And the assumption here, or the hypothesis, that if you really care about asthma, it is that neighborhood that you need to focus on, and you can kind of ignore the rest of it. Why is that relevant to have that neighborhood? Because we have 20,000 proteins in the human cell. There's no way we can actually explore all of them. We need the proverbial lamppost or the light, right, to kind of point to a certain neighborhood and say, that's where the action is. So we can actually take that neighborhood out, that subnetwork, which has now only a few hundred nodes, so I think in 300 in this case, and then we say, if you want to understand asthma, forget the rest of it. This is the neighborhood where the action is happening. And here we're kind of using simple graph theoretic tools to find that neighborhood. These days, of course, with the arrival of AI, we can also use AI-based kind of clustering methods to identify that. At the end, they all kind of give you the same, roughly, or same accuracy of identifying that network neighborhood. Now, how do we know that we are right, right? We found that network neighborhood. We find that statistically there's something happening there that you could not explain by chance, but is the biology cooperating with that assumption? And that's where kind of working together with biologists and experimentalists really kind of helps us go to the next level. We use network science to zoom into the network neighborhood, and now we use kind of biological information to check whether we are in the right neighborhood or not. And one way to say, in this case of asthma, glucocorticoid is a set of drugs that people kind of inhale if they have asthma. They're rather effective for asthma patients. Is it true that when you inhale glucocorticoid, then the genes that are being affected are those in the module and not outside of it? And the answer is yes. So we can actually our colleagues do the experiments. They take normal cells, they take asthmatic cells, they add glucocorticoid to each of them, and then we ask, where did the change happen? Did it happen in the neighborhood that we said that's where the asthma module is, or did it happen somewhere outside of it? And the answer in this case is strongly happens. And this is kind of the yellow lines, right? Whether it's normal or whether it's asthmatic, the changes are strongly happening in the neighborhood where we think the asthma module is. And the effect is even stronger for asthma, for asthmatic patients than to people who are normal. So for normal people, the effect could be a little bit more distributed, but for asthmatic patients, it's very, very well localized. And then what kind of value do you get out of that? Then you kind of start playing the biologist game to say, well, who are the genes now involved? Give me predictions that we didn't know about it, about the particular genes and proteins that are responsible for asthma. And we did indeed end up kind of identifying certain pathways that were not known before, like the GAB1 pathway. And then our colleagues at Harvard Medical School can go back and check whether we are right. And this is where the value of the network-based approaches comes, because what it means to check whether we are right. Well, it means that we're telling them precisely that it is these interactions and these proteins that are responsible for the effect. So if you kind of enhance the activity of one, we'll tell you who else should be actually changed. And they can explicitly do kind of a combinatorial experiment to check whether we are right. And in this case, of course, we are very much, the experiments have confirmed. Why am I telling you this story this way is that just to kind of tell you that this massive network data that really is the result of 30 years of experimentation, that we now conceptual of like who is interacting with whom. Now we conceptualize it as a network. And once we have a network, then the network itself provides us lots of new information about how the disease could work that eventually allows us to make very specific prediction about one or two or several proteins that can be tested experimentally. So what we call in physics or in biology, the predictions can be falsified experimentally. Now, what I showed you so far is a kind of a story around asthma. But what about other diseases? And here now in yellow, I'm highlighting another respiratory disease, which is COPD. As you know, COPD is often called the smoker's disease because you can get COPD even without ever smoking. But those who smoke actually have a much higher occurrence of COPD, and the vast majority of COPD patients actually are typically smokers. Very rare to have a genetic predisposition to that, but some do. And the symptoms are very similar to asthma. The mechanism of disease is different. And so interestingly, what we find is that the COPD genes tend to be in the vicinity of the asthma module. And this is not just a random coexistence. In general, we expect that diseases that have similar symptoms and hopefully similar mechanisms are located in the same network neighborhood. We can see that visually over here. How do we know that this is indeed happening? And for that, we had to introduce new measures, new graph theoretic measures to measure effectively the distance between two set of points on the graph. The two set of points in this case are like, say, the genes involved in disease 1 and the genes involved in disease B2. Like, for example, here I'm showing you three different diseases, one kind of labeled in red, the other in orange, and the other one in blue. So these are three distinct genetic diseases whose proteins have been associated by biologists. And the question is, which of these are overlapping in the network sense, which are close to each other and which they are not? And for that, we ended up inventing this kind of separation measure that simply measures the pairwise distance between the proteins of the same color, the AA or the BBs, and between the colors. And the way this separation measure works, that if two set of points in the network are overlapped, that they're in the same network neighborhood, they don't need to be in the same nodes, but they're very close to each other, they're in the same network neighborhood, then this measure is negative. And if, however, this measure is positive, it says that these two set of points are situated in two different neighborhoods of the network. And so now we have a measure to kind of measure the pairwise distances. So in this case, for example, for the orange and the red nodes, the measure is telling us we have a negative distance. And you see visually that kind of the orange and the red nodes are roughly in the same network neighborhood. but it is also telling us that the red and the blue are in the different network neighborhood, because SAB, the distance between them, is positive. And indeed, you can actually even visually see in the network that they are located in two different network neighborhoods. So now we have this measure, and now we can measure that for every pair of diseases. You can take asthma and cancer, or you can take asthma and COPD. And indeed, what we find, and this is kind of to make a long story short, we don't have to kind of go deeply into that, is that every time two diseases are overlapping in the network, that they're in the same network neighborhood, then they are very similar in terms of symptoms. They have the same relative risk. What does this mean? That if you get one, you're very likely to get the other one. So once, like there are lots of diseases that once you have one, then it affects the other one. Like, for example, diabetes and obesity are what we call comorbid, and so what we find is that diseases who are in the same network neighborhood tend to be comorbid. Those who are in the different network neighborhood, they do not show that particular behavior. And indeed, I can go ahead and kind of go ahead and look at every pair of disease. So here, each node corresponds to a disease. They are located wherever we find them in the network. Their size corresponds to how big the module associated with them is in the network. And now you see that some of them are overlapping, and those who overlap make sense biologically that they show high comorbidity, and those who don't overlap, they actually don't have that effect. So what is interesting about all of this is that I'm making lots of predictions purely based on one information, which is the network. That is how the different proteins are connected to each other. And that line of prediction now allows me to go back and tell you something about medicine and disease, and eventually down the line to actually go ahead and predict even drugs that could perturb that. And the reason why this works so well is because really fundamentally, the way the cell works is it works like a network, right? So we're not just trying to kind of conceptualize it as a network. It is working like a network, and these network type data actually are capturing those behaviors. So these and other observations have led to a field that is called network medicine today. And at the Harvard Medical School, there's a Channing Division of Network Medicine that has like about 200 researchers, mostly medical researchers, who are actually now using network-based paradigms to understand different type of diseases. And the tool set that provided by network medicine are many. I mean, one of the very interesting one that both AI-based researchers and graph theorists have worked a lot on is how do you find that neighborhood where a disease is residing? And as I told you, kind of 20 years ago, that was all network-based. Now it's actually lots of AI-based tools are being activated. Also, drug targets. Well, the drug has to hit within the neighborhood where the disease module is. So finding a disease module is the precondition actually to say, where would you perturb that? Like, where do you have to hit? So drug target identification has to be preceded with finding a disease module. Same drug combinations, very interesting questions come up is that when you have two drugs that are hitting the disease module, how would they hit? Should they hit in the same network neighborhood, or should they hit in a complementary network neighborhood to be effective, to be better together than any of them alone? And all of these tools are also offering ways to kind of think about personalized network medicine, which is kind of telling you actually whether for you a particular drug will work or a particular treatment will work. And I'll tell you in a second an example in that space. And finally, I'm going to spend a few minutes to talk about the last kind of example, which is drug repurposing. That is how this knowledge is being used to kind of identify whether the drug could work for another drug, for another disease. The question is, why do we care about drug repurposing? Well, because drugs are very, very difficult to approve, right? It takes 15 years to get the drug to the approval pipeline, typically, and about $1 billion. So once you have a drug approved, then you know what it does, and also you know the side effects. So it would be wonderful to know if it could actually cure not only the disease for which it has been approved for, but other diseases as well. And we have many examples for that. And so we ended up developing a drug repurposing methodology based on networks that came very, very handy when the COVID hit. Why was it handy? Because if you remember when COVID hit, now we know that the answer to COVID was actually the vaccines. But back then, when that happened in March, it was not at all clear that how long will it take to get the vaccines. Typically, to develop a new vaccine can last anywhere from 2 to 3 years to often decades. So we got lucky, but at that moment, the best strategy for us to kind of cure COVID was to find drugs that were already existing and used and approved and may actually positively affect COVID patients. So may help us cure actually somebody who is infected. So therefore, in March 2021, we ended up kind of forming a consortium that include researchers from Harvard Medical School, like Marinka Kicik, from my lab, a company that I funded, as well as MIT and BU, to apply the network medicine tools to see what of the existing drugs could actually work. And how do you conceptualize that problem from a graph theoretic perspective, right? Well, I already kind of introduced to you the concept of the disease module. But what is a disease module when it comes to COVID? Well, the COVID is an external agent, right? So it's a virus that injects itself into the human cell. But when it does so, it affects the cell by binding to some of the human proteins. So, and that list of where the COVID actually is binding was released early on at the COVID, that we know that there were about, if I remember, 200 to 300 proteins that were actually the COVID proteins could bind to. And we use that to identify what we call the COVID disease module. That is the network neighborhood in the human cell where the COVID proteins bind, because that's the network neighborhood that is being perturbed by the virus. And then the challenge became, are there drugs already out there who would affect that particular network neighborhood where that is being perturbed by the COVID? Because if there are drugs who can do that, then we may be able to perturb the COVID's ability to bind to the human cells and eventually to kind of develop the disease that we call COVID. So we ended up kind of identifying first the COVID neighborhood within that network. So imagine that purple place would now be COVID and not asthma. And then we're searching for drugs that hit in that particular neighborhood. Drugs that hit far away should have no effect whatsoever on COVID. That was the hypothesis. And we ended up doing a massive predictions setup where Marinka Kicik used AI-based tools that worked on the graph to predict what drug could be repurposed against COVID, knowing where it hits. And my lab, for example, used the network proximity, the one that I just kind of told you earlier, to kind of identify what drugs could again affect the disease. And then other people actually use some diffusion tools and so on. And within less than a month, actually, we ended up running all of these pipelines for COVID and coming up with a set of predictions of which of the drugs that are out there could have a potential effect on COVID. What are we talking about? There are about 7,000 drugs out there that are approved for human use. So the question, which of these 7,000 could actually have an impact of COVID? And for each of the drug, we know what are the targets. That is what human proteins are being targeted by that particular drug. So in about 3 weeks, we ended up making the set of predictions. And then the question, how do we know if we are right? And the way we knew we were right is that on one end, there were actually some drugs already in clinical trial because doctors believe that they should have an effect. So that's what we call the consensus knowledge. But we ended up pairing up with the BU Needle Center. If you know, BU has this massive high security center where they can work with very, very nasty viruses and pathogens in general, much nastier than COVID. And they ended up testing first our predictions, and eventually we together ended up, or mostly them, testing all 7 drugs against COVID. Of course, why we made our predictions in about 3 weeks? So the tool set was ready, we were done, and we closed the predictions. Then it took them about 2 months to actually get the first predictions. And then we said, are we right or not? And the answer was yes, we actually got many of them right, in the sense that our methodologies, network-based methodologies, rank them very, very high as potential repurposing candidates. And many of those actually turn out to be effective in the experiments as well. The experiments simply, you take a cell line and you actually add the drug, and then you infect it with COVID and say, did the COVID survive in the cell line or did it die? And the drugs, if it's effective, it should kill the COVID virus. It should not be able to infect it. Now, beyond the fact that we tested 7,000 drugs and we tested our methodology and the accuracy, one of the most exciting outcomes of that is that of the 77 drugs that shows efficacy for COVID, 76, that all but one, are what we call network-based drugs. So what is a network-based drug, right? Well, let's go back again and say, the SARS, for example, many of the drugs that were actually developed for COVID were meant to actually be binding to the COVID virus itself, that the proteins within the COVID virus, right? So that's what we call a direct viral drug. But then other drugs that turned out to be effective were not binding at all to COVID proteins, but they were binding to human proteins. And what was interesting is that the hypothesis that everybody was testing back then is that a drug would be effective against COVID if the drug hits one of the binding partners of the COVID proteins. So that when the COVID comes in and binds to a human cell, if you hit the same protein that the virus binds to, then you may have an effective drug. And we found that only one of the drugs that were effective against COVID had that property, that it was actually hitting one of the binding partners of the COVID virus. And the other 76 were just hitting in the network neighborhood. So you would not be able to identify them by simply knowing who are the targets. And these are what we call network drugs. And this is why network medicine is so important, because it turns out that about 9/10 of the drugs that we have right now on the market do not hit what we call the disease gene or the disease protein. They always hit in the network neighborhood of that. And so just knowing the disease gene is not enough to identify who your targets are. You need to do the full network. Why don't we hit directly the disease genes typically in biology? Why don't we kind of really knock out the node that is responsible for the disease? Because the node responsible for the disease typically has lots of functions in the cell, and you cannot mess with that, right? If you like shut that protein down, you're probably going to die for some other reason, right? So what you end up doing is that you end up finding what is the consequence of the problem in that protein, and you go downstream in the map and say, how do I fix that problem, right? In a little way, to kind of think about it, that if you have a traffic jam, one solution is to go and blast the traffic jam away and blow all the nodes, all the cars who are in that road up. The other is to go around it and find an alternative path. And really what the network does is that it says, I cannot deal with that mess, what's happening, because that's genetic and you're born with it. So I'm going to find a bypass and kind of affect the cell in a way that the cell does not feel the consequence of that problem that the defective protein causes. And for that, you must have a network thinking to understand what, to kind of unveil where would you hit, what network neighborhood do you want to hit. Now, so I will kind of conclude this part to say, on one end, like network medicine, on one end grew out of network science and graph theory. And it offers really a beautiful framework to how to think about the interconnectivity of the cell, but it does much more. It offers predictions that are now affecting patients, right? On one end, I already gave you the example of the COVID, right? And how many of the drugs we predicted, many of them actually ended up going on clinical trials and were used on patients. But another example which kind of comes back to personalized medicine is PRISM-RA. It's a blood test that I have helped develop, and a company that I funded it. So this is kind of, I'm a little conflict in it, but I'm still very proud of it, because this is a personalized medicine tool that, using network tools, we can tell you whether a particular drug is going to be effective for you or not. In this case, it's about rheumatoid arthritis or RA. for which the leading drug costs about $90,000 a year, and it's only effective to 30 to 40% of the patients, but it takes 6 months to a year to figure out whether it's going to be effective or not. You have to start taking the drug, and then 6 months later, you will see whether it did anything to you or not. And we ended up developing a blood-based test that looked at effectively the changes in your gene expression patterns in the network neighborhood where RA is related, and it can infer using an artificial an AI algorithm whether you will respond to the drug or not. So therefore, by just doing a broad test, you end up kind of not losing 6 months to wait to see whether the drug will work, never mind the amount of money you would actually pay for the drug during that period. And this is just the beginning. I think the future is really that for most drugs, we will start having tests. So first you do the test, and the test will tell the doctor, will that work for you? And many of these tests will be effectively network-based tests. Why do you need a network-based test and not just an AI algorithm to tell you that? Because you have too many parameters, right? You have 20,000 genes and you have tens of thousands of transcripts. So you need the network to narrow down what neighborhood you need to look at, what is the lamppost effect, right? And then once you have that network neighborhood, then you can use AI type of methods to find the combinations that really are predictive, whether you will respond to the disease or not. Now, as I said, kind of my lab has been focused in the last kind of 15 years or so, developing these network-based measures to understand human disease. But one of the things I was always frustrated about it is that everything that we do so far is based on genomics, right? Because at the end, who the proteins are genomic-based, what are the mutations that may affect their function genomic-based. But we know from our colleagues in genomics that genomics alone is not very predictive for disease outcomes. For certain diseases like Huntington's and so on, you got the mutation, you're going to get the disease, no way out, right? But for most diseases, you can be very predisposed to disease genetically and yet not get it. And heart disease is one of those, right? If you have all the mutations that predispose you to heart disease, all the negative effects you would get from the mutations could be actually changed by a healthy lifestyle. So environmental effects could really change the effect of the genomics. So I kept thinking, how do we actually incorporate these environmental effects into the network picture? That how does environment affect the network within the cell? And it turns out environmental effects on health are of multiple layers, like exercise is very important, stress is very important, but probably the biggest gorilla in the room as an environmental effect is really food, right? Is that because at the end, by eating, we are somehow perturbing ourselves on a daily basis. And the question I was really curious about it is that how does really kind of food perturb kind of the networks? And so to kind of come back to that, the question is, how much does environment and how much genetic matters? And it turns out if you take the top 10 reasons for why we die in the US, right? And you look at the genetic risk for them, you can only explain best case scenario 30% of the death from the genomics, right? So where is the rest? And the rest is all in the environment. And it's kind of very puzzling and also interesting that US is spending about 55% of its research budget on genomics, while really the contribution of genomics to health is typically about 10% for if you take across multiple diseases. So how does food affect health? Well, there's lots of evidence, particularly coming from nutrition, right? What you see on this map is actually the incidence of heart disease in the United States. And you see that even though the United States is considered to have a more or less homogeneous genetic background, thanks to the many mixings that we have, there are some ethnic differences in the South and so on of Mexican origin. But other than that, we have a European genetic background. But nevertheless, the incidence of the disease of heart disease can drastically change from one county to the next, right? And that should not be the case. And how important, for example, food and environment are is kind of shown very nicely by a study done on people of Japanese kind of origin, that if you are of Japanese origin and you live in Japan, then your chance of developing coronary heart disease is 1.6 out of 1,000. If you, however, with the same genomics, you move to Hawaii, that doubles. And if you go to San Francisco, that further goes up, right? So this is something that we all know. Environment really matters. And the question that we would want to ask is that how? How do you incorporate that environmental effect within the cell? And of course, at the end, food is molecules, right? And molecules at the end, we eat it, but eventually end up somehow arriving to our cells and feeding those cells. And that's kind of, we always think that the main role of food is to feed the cells. But I will also show you that food has other roles as well. It also modulates cellular activity in a way that is highly non-trivial. So we said, okay, well, if food is made of molecules, then we can go ahead and put it into this picture. And we just need to know what are those molecules that are in the food. And I thought, oh, this is easy, because about a few years earlier, we did a cool study that was kind of fun of its own, which is the flavor network problem, right? So this was totally independent of health. This was kind of motivated by the fun question of what food ingredients pair well together, like what should you put together in order to make a delicious meal? And this was kind of motivated by a hypothesis that at that time existed in molecular cuisine to tell us, well, the taste of the food is really determined by what we call the volatile components in the food. These are molecules that affect your taste buds, but mostly affect your nose. Probably it's obvious to all of you that our tongue and mouth can only taste about 4 or 5 tastes. That wide taste palette that we perceive is really through our nose, through volatile components. We smell the food, we're not really tasting it, right? And these kind of chemicals, these volatile components in the food, have been categorized by the food industry, because that's how they create these see-through actually liquids that you pour in the water, pour in your food, and it will taste smoky, or it will taste pepper, even though it's just a chemical, right? So these are these volatile components that are responsible for those tastes. And so we wanted to test, the hypothesis in molecular cuisine was that food pairing is a little bit like color matching, that the ingredients taste well together if they share one or several kind of food chemicals. That is that somehow we perceive them to be really tasting well together if there's something common between these ingredients. The alternative would be to be complementary, right? We try to put ingredients together that one brings certain chemicals and the other others, and then together they have some complementary effect and taste very well. So we ended up building this map where each node corresponds to ingredients, right? Wheat, egg, onion, olive oil, and so on. And 2 ingredients are connected if they have common volatile components. That is, that they have the same molecule that is responsible for some aspect of their taste. And we use this map to see whether that hypothesis in molecular cuisine is correct. And the answer was yes and no, depending on where you look. It turns out that European and American cuisine and Mexican cuisine is very, very much based on this shared chemical principle. Lasagna, for example, every single ingredient other than salt is sharing chemicals with every single ingredient within the lasagna. However, Asian food does the opposite. It's complementary. It's yin and yang, right? That you put things together that are otherwise have a different set of chemicals. But what this study kind of made me believe that, oh, we know what's in the food, right? Because this has been cataloged by the food industry. And that I was in a very, very big surprise. And the surprise really happened with that because we said, okay, well, let's just use this data. And then we realized that this data is really not capturing what's in our food. And let me just tell you a little story of how food affects our health. And this is one particular pathway that has been worked out by medical doctors and biologists. of why red meat is often not good for us to eat. And the molecular reason, one of them is the cholesterol, right? And there has been lots of papers about the cholesterol, so I'm not going to tell you that story. I'm going to tell you this other story, that choline, red meat has choline and carnitine. These are two molecules that is contained in red meat. If you eat red meat, you're eating these molecules, and these molecules are really processed by the gut bacteria, the bacteria within your gut. And from this, they create this other molecule that is often known as TMA that goes into your liver and becomes TMAO. And this is all healthy process, except that people who have, for whatever reason, an elevated TMAO level, they have 40% higher chance of dying of heart disease. So somehow TMAO became at least a biomarker, but potentially a causative agent. We don't know that towards heart disease. Now, the question is that why is it that, for example, Mediterranean diet says go ahead and eat red meat, and it doesn't seem to have that heart disease problem? As you know, Mediterranean diet is one of the healthiest diet out there, if it is not the healthiest. And there's an explanation for that, because the Mediterranean diet pairs meat with garlic and olive oil. And garlic and particularly and olive oil contain two chemicals that block the gut bacteria's ability to create TMA. So you can have the meat if you're pairing it with garlic or with olive oil, because you will not have elevated TMAO levels. Now, this is kind of a well-known story within biomedicine. What is interesting, I said, okay, fine, so we know how this works. How could I find out what food contains all of these molecules that may not be good for my health? And that's when I was in a real surprise. It turned out that of the 6 chemicals involved in this pathway, only one of them is being cataloged by what we call nutritional databases. The rest of them, you can find out who has it and in what quantity. And then we started digging deeper into that. And then we realized, wait, isn't nutrition's problem to kind of figure out what we eat and what are the chemicals in there? And then I realized that, yes, nutrition does a very, very good job of focusing on 150 nutrients. What are these? These are the energy sources that you must need to survive, right? These are the source of sugar, source of fats, and so on. And also some of the vitamins that we must have, because without that, we'll have vitamin deficiency and it will lead to disease. And that makes us believe that really that's all what food contains. But in my database now, many years later, we actually curate a database that has 135,000 distinct chemicals that are carried in food. And this is what we ended up calling the dark matter of nutrition, because outside of this 150, this 135,000, you have no idea what food contains how much of it. These are not being measured systematically, not being cataloged, and so on. Let me give a specific example. For when it comes to garlic, it has 67 nutritional components. This is known thanks to nutrition, but has more than 5,000 other compounds that are not nutritional in the sense that they don't provide energy to us, but they are essential, many of them, to our health. 2/3 of the molecules that are kind of in this list had known health effects, in the sense that molecular biologists have shown that they affect certain cellular properties. So now we're facing with this dark matter of nutrition, right? That really there is this unmet chemical complexity that is in a diet, and we wanted to ask, how do we understand its effects on health? And just to kind of be specific, for example, if you say what basil is, right? Basil consists of more than 2,000 different chemical compounds, of which 146 are kind of quantified, that we know how much. The rest, we don't know how much is it in there. And many of them are responsible for flavor. About 300 are known to be disease-related, right? So these kind of like, if you have too much or too little, could lead to disease, and others have other health implications and so on. So this kind of prompted us to start in my lab the Foodome project, which is we tried to catalog what are the molecules in the food. That's why I said 135,000. But the most important thing that I would like to share with you in the rest of the talk is to say, how do these molecules affect our health through kind of like somehow affecting this particular network? And to understand that, I'll tell you one story that is focused on polyphenols. We all know, you probably all heard of polyphenols, and you heard that that's good for you, right? And you probably also heard that polyphenols are typically in plants. They're generated by what we call the secondary metabolism. They believe to be antioxidant and lots of good stuff. So that's one of the reasons why always you're being told that eat fresh vegetables and fruits with your diet, or have your diet be mainly based on fresh vegetables and fruits, is because they are rich in polyphenols. Now, the question is, what do polyphenols do within your cell? And if you are tempted to think that they are like being processed by the metabolism because they provide energy or something good to us, that's what I thought when I came to this research as well. It turns out polyphenols are not processed by our metabolism. That is, that they are not chemicals that our cells take up and they're a source of energy or something else. Rather, they are providing chemicals that bind to the human proteins, and they regulate the activity of that. So they bind and they block certain activities, and through that binding, effectively, they act like drugs. And the beneficial effect of the polyphenols is coming not from their source of anything in particular, but their ability to bind, to modulate cellular activity. So that's when we said, how do they do that? And to kind of give you an idea from a network perspective, what it means is that, let's take green tea, right? Green tea's active ingredient is one particular polyphenol that has this very long name, and we just called it EGCG, right? It's a well-characterized chemical. And it turns out that this chemical, the green tea chemical, can actually bind to 52 human proteins. That is, once it gets in your bloodstream, it arrives to your cell, and it finds one of those proteins, it connects to a certain place within that. And so let's say that this is where the polyphenol targets are. And we would like to understand what kind of diseases could drinking green tea actually affect. And for that, we ended up putting on this network also the disease modules. We already introduced the concept of the disease module for all diseases. And the hypothesis is that if the polyphenol targets hit in the vicinity of one particular disease module, it may modulate that disease, like it may have beneficial or sometimes negative effect on that disease. So that's kind of the hypothesis. So we ended up putting the targets on this network that we did earlier and say what diseases are in the neighborhood of the polyphenol targets. And in this schematic one, we would expect that disease A would be affected, but disease B and C would not be affected by drinking green tea because they are too far away in the network. And we did that, and indeed it turns out that there is a network neighborhood where the EGCG targets are located. And one of the diseases that is located in that very network neighborhood is type 2 diabetes. And so the prediction would be that it should have effect on type 2 diabetes. But we ended up using this network-based ranking to say, let's rank all diseases, whether based on the network only, purely on the network, we think that green tea should be affected by that, should affect that disease. And when we did that, these were actually the diseases that we ended up ranking kind of from the top to the bottom. And it turned out that the top 10 were all known to be affected. There was previous clinical or observational data to be affected by green tea, and the bottom ones were not affected. So that was very, very exciting that we ended up saying, hey, the tools are working. We're ranking high those that are known to be affecting. But we actually went much further. These are actually 4 different polyphenols, and we made specific predictions how it is actually affecting the cell and how is it contributing to disease. And for example, we connected each of them to different diseases, and I'm going to focus on rosmaritic acid, where we said it affects platelet activation, which is a very important process in heart disease. And we took that further. We made the very specific prediction of how it should affect platelet activation. And then, of course, my colleague, Joe Loscaso, and his team at Harvard Medical School ended up doing the experiment and experimentally confirming that really kind of it does affect health platelet activation. Again, I'm just showing you how this final example to see how we can go from this very, very large big data like network data, and combining with the right information, we can make specifics that are accurate at the level of the individual proteins. And this is where the power of the network is, right? And we're not at all surprised, and I was not at all surprised that technology does it on a daily basis. When I left from Brookline, I totally relied on Google Maps to tell me how I'm going to arrive here, right? And I did arrive here. And a similar thing is happening now within the cell, right? And within network medicine is that these maps are becoming accurate enough that they act like Google Maps and give us the ability to make predictions that can be experimentally tested and eventually can be brought to back to us as treatments or diagnosis tool and so on. So these days we have actually this kind of Foodome project in my lab that kind of does many predictions that we're trying to kind of taking back to the patients, and slowly my time is out. So I will kind of end here. And just to kind of give you the big picture, I'm a big believer that if we want to understand life to begin with, and then the breakdown of life, which is the first manifestation is disease, it's not enough to know the components. You need to know the network, right? Because the way this system works is driven by the network. And you can take this analogy to other systems as well as we do in the lab. You can take it to social systems, you can take it to economic systems, to technological systems. The components, knowing the components take you only that far. Really, you need to have the full network to understand how they work. And within medicine, I am convinced that 20, 30 years from now, it's not enough for you to be a good cardiologist or the good neurologist. You're going to have to be a good networkologist as well to provide good treatment to your patients. And I will end actually by acknowledging the many collaborators who have helped us do this. And there are many more, and happy to take any questions that emerge. Thank you.